Hi, everyone. I guess we'll go ahead and get started here. My name is Nick Langholz. I'm the Program Director for Neural Engineering at the National Institute of Neurological Disorders and Stroke here at NIH. I'd like to welcome you all to our third Brain Initiative webinar on technologies for recording and modulating neural activity. The overall structure of the presentations today is we'll have about a 20-minute presentation from our two invited speakers, followed by a 10-minute discussion period. That 10-minute discussion period, we have pre-selected discussants, which are Bernardo Sabatini and Li Hong Wang. Beyond that, there isn't necessarily an open period for questions, so hopefully whatever questions you may have would be answered by, or would be addressed by those individual speakers. Otherwise, we're hoping to have the ability to have offline questions to the individual investigators should you have them after the webinar. These webinars will also be recorded and placed onto a website to allow for offline viewing. So without further ado, I'd like to go ahead and introduce Changhuei Yang to present on time reversal optical focusing for optogenetic activation. I hope you enjoy the presentation.
All right. Thank you, Matt. So I just want to thank the organizers for giving me this opportunity to talk to you today. So what I'm going to do is sort of go through the project that Viviana Graninaru and myself is doing for the Brain Initiative effort. And what we're trying to do really here is to actually create a method by which we can actually do deep brain stimulation for optogenetics purposes using this time reversal or wavefront engineering approach. So let me get into it by talking about the fundamental challenge that we're trying to address here. So I don't think this needs a lot of explanation. The brain, the reason why it's actually very difficult to work with the brain using the optical light field is the fact that tissue is very turbid, in the context of the optical wavelength. So that tissue turbidity really confounds our ability to actually reach deeper into the brain using optical methods. And the reason, the way that people have been actually trying to address this tissue turbidity has predominantly been to use longer wavelength. The longer the wavelength that you go, the less scattering that you would experience, and thereby allowing you to actually reach deeper into the brain. But unfortunately, that strategy do have a limit associated with it, which is the fact that as you go for a longer and longer wavelength, you eventually reach a point in which the absorption due to water starts to actually limit your ability to fundamentally deliver light into the tissue. So what we're trying to do here is to actually come at this with a different strategy in which we are not trying to actually minimize the scattering by choosing to use a different wavelength, but to actually try to correct for scattering on the way in so that we can actually access deeper parts of the brain by correcting the scattering on the way in. So we have actually demonstrated that, for a piece of tissue, that you can actually use this kind of strategies that we're talking about to actually access at a depth that's beyond the scattering mean-free path, which is typically about 100 microns, and actually beyond the transport mean-free path, which is typically on the order of a millimeter. And fundamentally, this kind of method is really sort of bounded in terms of its depth penetration by the absorptional characteristic length associated with the tissue. All right, and that typically ranges on the order of tens of centimeters. All right, so I just want to basically clarify what we're trying to do here. We're trying to basically create a situation in which we have a wavefront associated with a light that goes through the fog or a scattering medium, such as brain, in such a way that it actually comes to a focal point on the other side or in the middle of the fog. So if you think about this, right, this is a pretty strange wavefront that we'd actually have to create, because it would actually have to come to a focal point within the fog itself. So that means that we need to do something that is quite unconventional. Right. Now, if you can indeed actually create such a focal point of light within the fog or on the other side of the fog, then we have a very flexible tool. Because you can imagine if you take that point of light and raster scan that across your sample of interest, you can potentially perform imaging for fluorescence image collection or for Raman data collection. You can also think about using this focal point of light to do ablation, because all you need to do is turn up the light field power, and you basically have a tool by which you can start to ablate tissue very locally. And then finally, you can think about using that focal point of light to do optogenetic activation, which is basically the objective that we have currently. Okay. The way we do this, actually, we have to actually make use of the fact that scattering is actually time symmetric in nature. Even though, when we typically think about scattering, right, it looks like a random process. Yes, it does actually create a situation where light that's coming out from the other side of the scattering medium is indeed random, but that randomness is deterministic in nature. And because of that deterministic nature of scattering, you can actually exploit it to do something very interesting. So I have here a basic illustration of what would go on when you have light interacting with scatterers. As the light passes through the scatterers, it gets disordered on the other side. But if you're actually able to take that light field and create a time reverse or conjugate copy of the light field and send it back through the scattering medium, it would actually be able to interact with the scatterers in a reverse fashion in such a way that it would actually allow you to recover back the original light field that you sent through. And this is what we mean by the fact that scattering is time symmetric in nature. All right. You may think that to actually create such a situation, it must be pretty tough. Yes, it is tough if you're talking about water wave. But in the optical domain, we actually have a class of methods that allows us to do this kind of conjugation or time reversal playback in an effective fashion. Holography is one approach by which you can do this. And this whole general class of method, we call it phase conjugation. All right, so I kind of want to walk this through with you a little bit so that we can appreciate what is going on. So imagine if you have a point source of light and it's actually impinging on a mirror, then you expect that light to be reflected in a way that we typically can understand and appreciate quite well. But if you replace that mirror with a phase conjugate mirror, or this time reversal plane, then what is going to happen is that as the light is reflected off from this interface, it's actually going to create a wavefront that would then act as if it was traveling backwards in time and be able to retrace its path back to its source itself. Mathematically, what is going on here is actually fairly simple. All that's going on is that the wavefront that is at that interface, what that interface is going to do is it's simply going to take that wavefront phase at each point on that interface and simply flip its sign. All right, so if you do that, you basically would have created this phase conjugate copy of a wavefront or a time reverse copy of a wavefront that can behave as if it would travel backwards in time. Okay. All right. So holography is one approach by which you can do create this. Subsequently, I'm going to talk about a more efficient approach by which you can actually generate this conjugate wavefront so that we can do practical things in tissues. All right, again, I just want to draw the attention back to the fact that what we're really trying to do here is to be able to create a focal point of light on the other side of this scattering medium so that we can actually do useful things with it, okay, or in the middle of the scattering medium. And the strategy that we're employing here is known as time reversal ultrasound encoded focusing. And this proof of concept was actually first demonstrated by Li-Hong Wang back in 2011. So let me just walk you through very quickly what this approach is all about. So first thing that you want to do is you basically want to take a piece of tissue represented by this pink square here, and basically put light through it. This light is going to scatter every which way as it passes through this piece of tissue. And then what we want to do is you want to turn on the ultrasound and focus the ultrasound somewhere deep within that piece of tissue. Ultrasound, unlike light, is not going to be scattered significantly by the tissue, so it's able to actually render an ultrasound focus somewhere within that tissue quite effectively. Then what's going to happen is that as that light passes through this piece of tissue, some of the light is going to pass right through that ultrasound focus. And when it does that, part of that light is going to be upshifted in frequency by a amount that is exactly equal to the ultrasound frequency. So on the exit side, what you have coming out is basically going to be a light field that is unmodulated, and then a light field that has been upshifted in frequency. And that upshifted frequency light field is uniquely associated with that point at that focal point of the ultrasound beam itself. And what we do here is that we basically selectively collect the wavefront that is associated with this upshifted component of the wavefront. And by this time-symmetric nature of scattering, if we play back a conjugate of time-reverse copy of the wavefront, this light field is going to be able to retrace its path back through this piece of tissue and render a focal point, optical focal point, where the ultrasound focus is originally at. So this is the way by which we can actually generate a situation in which we can focus light to a point within the scattering medium, even if the tissue depth here can exceed, the transport mean free path associated with that piece of tissue. Okay. All right. So we've gone through a few concepts, and I thought it'd be worthwhile to systematically, sort of present to you the sort of experimental verification of all of this stuff. So the first question that we should address is tissue scattering really time-symmetric in nature? So back in 2008, we actually demonstrated that concretely that indeed this phenomena is actually experimentally observable. So let me walk you through very quickly this experiment. What we have here is basically a light field that has a certain pattern to it. If we put that light field through a piece of clear agarose, and if you put your eye on the other side, you can see a very nice transmitted light field pattern that's associated with the original light field itself. Now, if you now replace that clear piece of agarose with a piece of tissue, you can see that on the transmission side, the light field pattern no longer look crisp and clear. Instead, you get this very blurry pattern because of the fact that the light has been scattered by the piece of tissue. Now, if you now take that light field and you record that and play back a conjugate copy of the light field, and if you now put your eye on the other side, you can see that we can actually observe a fairly clear and crisp reconstruction of the original light field that we sent through. So even though the light has now passed through the tissue twice, and by that effect should have been scattered twice, you can actually get back a pretty nice reconstruction of the original light field. All right, so this demonstrates the fact that this scattering is indeed time-symmetric in nature, and yes, you can indeed use a conjugation approach to actually undo the effect or correct the effect of scattering. All right, we actually did a series of experiments to demonstrate that you can actually use tissues of very high thickness and still be able to actually elicit this phenomenon. We have actually tried tissue all the way up to about 7 millimeter at a wavelength of 532 nanometers. So at that wavelength, the average scattering events per photon is over 200 scattering events. And yet you can see that we can readily elicit the phenomena and create a reconstruction of a focal point of laser beam that we put through that piece of tissue. And another thing that's worth noting here is the fact that you may think that for us to do this kind of correction, we've got to be able to record a very big fraction of the scattered light field. In fact, that's not the case. Even if you collect only a small fraction of light field, you can still actually elicit this phenomenon. In this particular line of experiments, we only collect about 0.02% of the wavefront, and yet we can still observe a pretty nice reconstruction. Okay, another thing that we need to actually implement to make this method practical is that we need to create a time reversal or phase conjugate mirror that is able to actually render a much stronger playback or conjugate wavefront than the initial light field that we're trying to conjugate. Why do we need that? Well, if you look at what is happening here, ideally during this recording phase, the wavefront that we use ought to be a weak one so that we're minimally perturbing the tissue. And then once we have done the recording, ideally when we do a playback to render the focus, we want to be able to render a very strong focus. So the light that's actually being generated by this conjugate device here needs to be very effective in terms of rendering a strong optical point here. All right, so you need a weak signal for recording and a strong signal for playback. All right, so conventionally, if you think of this as a piece of mirror, then it wouldn't cut it because if you think of a mirror, that reflection is a weak reflection. So we need to do something a little bit different here. All right. Now, up to now, right, the kind of experiments that we've been talking about has typically been done with photorefractive crystals. And photorefractive crystals actually have a pretty low reflectivity associated with it, as well as other disadvantages, such as the fact that it doesn't really allow you to manipulate the wavefront that you're doing the playback with. Neither does it actually allow you to use a full range of wavelength that you might want to flexibly change to for use with that piece of crystal. To actually address this, we actually developed this method called the digital optical phase conjugation approach, by which we can actually do a much more effective and efficient playback of the conjugate wavefront. So schematically, it's actually fairly simple. What we do is we just simply take the input light field and interferometrically detect its phase front on a CCD camera or any kind of camera. And then what we do is we take that data, we process it, and then we render a right pattern to put on a spatial light modulator. Then we come in with a blank laser beam that is then reflected off from this spatial light modulator to create this conjugate wavefront. All right, so with this approach, you can see that now we can actually increase very significantly the optical phase conjugation output as we desire, simply by cranking up the power of this blank laser beam that we're reflecting off from the spatial light modulator. This approach, we have been working on it steadily since 2010, and thus far, our capability is that we can actually do this kind of rendering and playback with a reaction time of about 5 milliseconds. And we can actually control a number of optical modes that is on the order of 10 to the power 5. This optical mode, let me explain a little bit why it matters and how does it impact on our experiments. All right. So in effect, right, what we're trying to do here is we're really trying to find the right wavefront solution so that if we look at the contribution of each of the phaser that's associated with the light that's coming in, at the points of interest, they all line up in a constructive fashion so that we can get a nice peak versus the background. The background is basically going to be dominated by simply the light that's diffused through this piece of tissue. And if you look at each of this point, right, the light field phaser itself is simply a combination of random walks from the contribution from the input light field. So the conjugation process allows us to actually find the right wavefront so that we can get this nice constructive interference of all the phasers at the point of interest. Now, how strong you can actually create this peak to be is actually dependent on how many points of controls you have on this input side. So the more points of control that you have, the stronger the peak. And that actually relates to that optical modes of controls that we were talking about just now. So that 10 to the power 5 optical modes of control that we were talking about basically relates to how many points that we can actually control on this input side to get to this construction. So with 10 to the power 5 optical modes of control, if we actually focus to a single point of interest, that can get us about 10 to the power 5 in terms of its peak strength versus its background. Okay. All right. To do this kind of experiments, of course, we need to be able to adapt our method to work with living animals. Now, in living animals, for this reversal to actually occur, we've got to make sure that the scatterers remain more or less stationary while we're doing the experiments. So it's actually very important and relevant for us to figure out what kind of time constant are we talking about when we're doing animal experiments. So this is actually one of the recent experiments that we've done to actually demonstrate that, for a living animal, we can actually still elicit the phenomena quite nicely. So what we have here is basically a living rodent that we have pinched up the skin. And what we did is we simply send a laser beam through it and look at the speckle pattern on the other side. So here's the speckle pattern. We take that speckle pattern and we conjugate that and play it back through this piece of tissue. You can see a pretty nice reconstruction of the original laser beam itself. Turns out, if you actually look at how long this ability to do a conjugation persists, it can actually persist over tens of seconds, as long as you can actually stop blood from flowing through this piece of tissue. And if you actually relax that piece of tissue so that blood is now flowing, the time actually drops to on the order of sub-seconds in terms of its decorrelation time. All right, so this means that if we're actually able to do experiments, hopefully in the milliseconds time domain, we should be able to create a situation by which we can still do this kind of phase conjugation, retracing of scattering, scattering and reversal. Okay. We recently demonstrated that, with a free piece of skin pinch, if we get our response time down to 5 milliseconds, we can actually indefinitely maintain this focusing through a piece of tissue over an indefinite amount of time, as long as we can actually correct for the wavefront solution over that 5 millisecond time scale. All right. So this gives us good hope that the kind of experiments that we are actually going to pursue with the brain is going to work. All right. Just want to show you our initial experiments that was done a few years ago in terms of actually implementing that kind of true focusing approach. Okay, so this is actually an experiment that was done using that high efficiency digital optical phase conjugation apparatus, and we're actually able to show that we can indeed focus light through a thick piece of tissue quite effectively. Here's a phantom showing you the kind of focusing that we can achieve. So we have basically two pieces of scattering medium here, and right in the middle we have a piece of fluorescent light sheet, sheet. And you can see that if we actually simply send light through this piece of scattering tissue and not do anything, the light is actually quite scattered as it passes through that piece of tissue, and we can actually experimentally visualize what is going on through the use of that quantum dot sheet. Now, we turn on this optical focusing approach. You can see that it actually can come to a focal point that coincides with where the ultrasound focus is. And then we can actually, by repeating the experiment and moving the ultrasound to different locations, we can actually move that optical focus to any point within that volume quite effectively. Using an Abbott method, we actually was able to show ability to do fluorescence imaging with a resolution of about 30 microns through a piece of tissue that's 5 millimeters thick. Okay, so we have been actually adapting this scheme for use for optogenetic activation. Our current experiment is actually to simply show that we can use this method to optically activate a opsin tag neuron through a piece of tissue through a piece of brain slice that's a millimeter thick. So we're currently doing an experiment where we're actually trying to do all the probe and focusing from one side, and then use a electrode to actually detect the electro signal on this side. All right, so this is actually currently work in progress. Our aimed spot size is about 60 microns with a tissue thickness of about 1 millimeter. This is actually a demonstration of what we have been able to show so far. We have yet to actually move our ultrasound to the other side, but the true system is currently on just one side. You can see that we are actually able to focus light through that millimeter thick brain section, and was able to use a camera to look at that focal point while it's formed. Here's a video showing what we can observe on this side. So when we turn on the true focusing, you can see indeed we can focus light through that piece of tissue. And when we turn it off, the background, it should just be the background. So you can see this is just the background when the true system is not focusing at the appropriate point. So at this point in time, the true engine that we're using for this particular experiment has a response time of about 280 milliseconds, and the peak to background associated with this focus spot size is about 10 micron. It's about a factor of 10. So that's basically where we are at this point in time. We're hoping that we'll be able to progress to an intact mouse brain experiment where we're using this method to actually go a few millimeters into the brain and do a non-invasive activation. Currently, that's a couple of challenges. This tissue decorrelation time from blood pulsation and other motions is something that we worry about and is trying to actually address. The other thing is that there's actually a limited amount of working space for a lot of equipment that we need to place around the rodent's brain. So we're actually working hard on actually compacting everything effectively enough so that we can actually do this experiment going forward. All right, so there are some compelling advantages that I would like to just highlight in this last slide regarding this, which is the fact that this approach will provide us with a deep, non-invasive optical focusing approach that allows us to actually do optogenetic type experiments. So in the long run, it can potentially be significant for non-invasive human brain work because it doesn't involve the use of invasive probes. You can also think of using this as an ablation tool. And then finally, it can be adapted for imaging and optical analysis beyond simple activation. Okay. So with that, I just want to acknowledge a group of very hardworking and talented students and postdocs that's contributing to this particular project. Thank you.
Thanks, Changhuei. And at this point, we'll turn it over to Li Hong to start the discussion period.
All right, thank you. First of all, before I ask the question, I have to disclose that my lab is actually moving to CalTech, so Changhuei and I will be colleagues. If my questions appear to be biased, you understand why. Changhuei, excellent talk. You talked about motion, right, due to blood pulse.
Yeah.
Most of the time we deal with the random motions that would decorrelate the speckles. But if there's some sort of synchronized motions and the scatters are moving synchronously, I wonder if we can maintain speckle correlation.
Yeah, so that's something that we observe in that slide that I presented just now. So you can see, oops, how do I go back? So you can see that in this particular movie itself, the reconstructed spot actually comes back after it has been randomized, has been decorrelated, and this motion that you're seeing here is actually because of the breathing motion of the animal pulling the skin. So this kind of motion, I think we can actually synchronize over a much longer time scale to actually maintain the focus. The motion that's actually associated with blood, though, is likely to be unsynchronizable, meaning that path that pass right through the blood vessel, there's nothing we can likely do for us to actually get to a point where we can correct for that. But the good thing is the fact that the blood pulsation, right, actually really do two things. So the blood itself that's flowing in the blood vessel, there's no hope for us to actually do a time reversal through it. But there's actually another motion, which is that blood pulsation is momentarily disrupting the scatter configuration of the other parts of the tissue that doesn't have blood in it. That motion ought to be reversible once the blood pulsation comes back to its same cycle. So that motion, I believe, we can correct for it going forward.
Right, right. So some time ago we observed two time constants for speckle correlation. One drops like a rock, that's really fast, probably related to blood motion. The other one has a much greater time constant, due to the slower motion. Right. So I presume we can always take advantage of the greater time constant.
Yes, I agree. Yeah. So we also have been doing experiments where we're looking at the speckle decorrelation time in a living intact mouse brain. And one of the things that we are facing challenge with is the fact that the brain itself is kind of like a jello, so it's actually sort of quivering within the skull itself. So one thing that we also actually like to do going forward is to actually put in a cranial window to try to pin down the brain to some extent while we're conducting experiments. So that hopefully would also help to actually immobilize the brain significantly enough.
Right.
So we'll see what happens.
Yeah. A quick question. This wavefront technology has a conference of Photonics West. Within the past few years has grown to be one of the largest. It's probably the fastest growing conference. Where would you project the future of this technology for the next maybe few years?
I think Certainly, the field is growing rapidly. There's actually a very interesting physics question that can be answered. So there's part of the wavefront community that's addressing that. But practically, I think there is really a lot that we can do. Already, we have shown that you can actually exceed the transport mean prepath in your ability to actually do focusing through tissue, and that is significant because thus far, we always have been limited by our traditional optical methods in terms of tackling scattering. This really tackles scattering in a way that directly addresses the issue at hand, rather than trying to go around it. And there are actually methods that are currently in development that makes use of this for other applications. For example, you can actually use this to also do a microscopy in a way that actually makes use of the scattering to actually improve your resolution. So again, I think that is an area that is interesting and worth looking into.
Right. Great. Thank you. I think I should give Bernardo a chance.
Yes. So I'm thinking about it in terms of a potential user. It's really quite beautiful technology. So at what point will we reach a fundamental limit from absorption? And what is the theoretical limit due to the acoustic focusing?
So you got a good point. The acoustic focusing likely is going to impose a limit sooner than the absorption from the optical wavelength at the optical wavelength. Right now, the shorter the ultrasound wavelength, the less penetration that you get. I expect that we can practically do experiment with ultrasound to a range of maybe 5 millimeter to a centimeter before the efficiency becomes impossible for us to push with ultrasound methods. But the good thing is that you're basically using ultrasound here as a way to tag the light. So there are actually other strategies by which you can actually tag the light, and those might give you actually a better penetration compared to ultrasound. So Li Hong and my group, for example, we have been using motions, intrinsic motions of things within the tissue to actually tag the light too. So that actually represents one other approach for tagging the light. You can also potentially use methods such as using ultrasound microbubbles. You can inject tissues with microbubbles and use the destruction of those microbubbles using ultrasound methods or optical methods as a way to actually tag the light as well. And that, again, represents other paths by which can go potentially deeper into the tissue.
Right. Should we?
Well, thanks, Bernardo and Li Hong and Changhuei as well. At this point, I think we'll go ahead and move on to Euisik Yoon and Yuri Buzaki for their presentation on towards high-density optoelectrodes integrating light onto Michigan probes.
Thank you, Nate. Do you see my screen?
Nope, not yet. You may need to go back up and redo the share again.
Okay.
Yep, there we go.
All right. All right, thank you, Nick. As compared to Changhuei's approach, we have been working on optogenetic probe more invasively, putting light onto the Michigan silicon probe. And this presentation, let me talk about the work that we do under current U01 grant, also in conjunction with the another R21 grant. All of the three, me, myself, and Ken Wise and Yuri Buzaki are co-PIs. As you can see, there's an evolution of the Michigan probe from passive recording array into making high-density recording site onto the region, and by integrating a circuit monolithically, also hybrid approach in a different configurations. And the question here is, this one can have a large density electrical recording, also electrical stimulation. But the recent advancements of the optogenetics, we wanted to incorporate optical stimulation capability onto this silicon probe. And as you can see here, optogenetics can allow us having a cell type specificity as well as a high spatial temporal resolution However, most of the chronic recording in neuroscience community is still using optical fiber sticking onto the brain and the flood exposure of the light into the whole region of the brain. And the question here that we wanted to address here is, can we deliver light to the specific local area of the target brain? So one of the approach, the way is you can put a tetrode or wire surrounding optical fiber and putting here the 4 recording sites along the optical fiber, and still optical fiber is large and also limitation of the recording site. And the other approach is actually done by Stark and glue a thin optical fiber onto the silicon probe. As you can see, you can have a multiple optical fibers thinned down to 6 to 70 micron attached onto the probe. It has a beautiful result. However, this is also having a manual assembly process, lack of accuracy. As you can see, the distal end is not really well aligned and a bulky and having a low yield. And when I take a look at this, and this is very intriguing, and from MEMS engineer, we can help out this by integrating optical waveguide directly onto the silicon probe. This is a approach that we took, and the monolithic integration of the SUA polymer waveguide on the probe shank. That's our initial device, and the light coming from the optical fiber from the back, and through the MEMS and etching technology, we can make a step alignment of the optical fiber to the optical waveguide in the back end, and the light is coming out from the tip in the distal end. We can exactly position the light illumination with respect to the recording site like this. And we can have a pretty good precision because everything is defined by photolithography within ±1 micron. And fabrication process is pretty simple, standard. I'm not going to go into detail. This is a typical Michigan silicon probe fabrication technology. The one exception is that we can put down a PECVD cladding layer at the bottom and putting a optical waveguide defined on the top of that. It is a polymer waveguide. It works pretty well. However, for chronic recording, we thought it would be better to have a dielectric waveguide with the cladding layer on the top. And this is the second generation of the waveguide probes, and having a oxynitride as a core layer with the oxide cladding. As compared to a SUA waveguide, we can reduce the intrusion of the tissue by reduce the size of the optical waveguide height from 15 micron to 5 micron. But however, with this small geometry, we still can have a high efficiency delivering power to the light source from the back onto the distal end at the silicon tip. And we put this into the animal in the Euisik lab, and this animal has a channel rhodopsin expressed neurons, and it has a single unit activity spike monitored along with the optical stimulation. As you can see, this red mark, these neurons are well synchronized with the optical stimulation, while you can see this pink neurons denoted over here has a little bit of the latency. And the next slide, it can show the better characteristic, this temporal relation between optical stimulation and induced spiking. And this red neuron cells, which is around 70% of the neuron, has a short latency of 4 milliseconds. So this thing has a very direct light-related activation, while the pink cells has a delay latency of 40 milliseconds, which has an indirect. You can talk about a little later. This is the indirect network-driven effect. The next question is, can we do a little more, and can we making a lot of illumination sites by having a splitter integration from the single source. Still, this is optical fiber. We can split the light into the 2 different shanks, and the light is coming out at the end. And also, we can make many different variations and The light can be coming out from the at the end or in the middle, and the multi-shank light illumination from the same source. But there's no control over each illumination site individually. The other one that we worked on is integrating optical mixer, which will give us a opportunity to incorporate two different wavelengths. And this optical wave mixer has a two light source attached, guided with the optical fiber. And then by switching the optical light source, we can illuminate the blue light for channel Watson stimulation, excite the neurons. And here is that we can switch to the yellow light, which will excite the halorhodopsin to inhibit the neurons. So we could successfully integrated optical mixer and then switch on the light. But still, light source coming from the source through the optical fiber and causing a big tethering problem for chronic recording. The question here that we wanted to answer is, can we actually integrate the light source in the back end of the probe? It is pretty doable because of blue, thanks to this electronic industry. You may have one of these light sources at your home, and it's a blue light. It's a laser diode, which will illuminate the blue wavelength. The question is, how can you attach on this laser diode at the back end with the high coupling efficiency? The idea here is we can use a GRIN lens, which is the gradient index lens, which has a gradient of the index. So when you have a point source light from your laser diode, even though you have a little bit of misalignment, which can allow up to 20 micron, with this we can have a good assembly with the high yield. Light will be guided onto the center, coming up from the center of the other side of the GRIN lens. So by having this one, we have a pretty good tolerance of the assembly, so we can have a high yield to assemble multiple sites and multiple light source illumination. And this is the schematic of the structure, and this is the typical Michigan probe with the optical waveguide integrated on top. And this is the jig that we made through the micromachining, etched the hole, that we can place the GRIN lens. And this is the LD, and we put the two LD. One is the blue, the other one is red, 635 nanometer and 45 nanometer. And once we align the probe with the waveguide and the LD in the back, we can place the GRIN lens through this etched hole, then we can have a pretty good satellite assembly process. And one of the other advantages of this GRIN lens is that it provides a thermal isolation. Silicon is a pretty good thermal conductor. If you're putting a butt coupling of the LD light source in the back, and you can actually increase the temperature of the probe. However, if we're having a GRIN, they will give us a thermal isolation from the illumination side of the source to the probe tip, so we can have a pretty good thermal isolation and then minimize the temperature increase at the tip of the probe. And this is a fabricated, the first prototype device. We integrated LV in the back. This is a cap to cover the illumination site, and this is the one that you can see. This is the probe, and this is actually not optical fiber; it's a GRIN lens, and the LVs are sitting in the back. And this is the probe tip, and we have a recording side, and this is the end of the with optical waveguide. And you can see by switching the LV, you can have a blue and red, and also mix the color. You can switch in with a very high spatial temporal resolution. And this is the in-vivo recording data, and the first one is no light, and we can have a spontaneous response of neural activity. This is not the process data, it's a raw data. Once you're having illumination of the blue of the channel rod of SIN, then we can have a stimulation of interneuron. As you can see in this probe, these are the interneuron with the channel rod of SIN will be excited. However, in its 635 nanometer, and the red light is coming, and it is pretty inhibit the neural activity through the pyramidal neuron. We didn't use halo rhodopsin in this case, and Yuri can talk about a little more later. We can use arch rhodopsin for inhibition and using a channel rhodopsin for the activation. The question here is the next one, which is not under current U01 grant, but related to R21, is actually, can we really making a probe so that we can integrate light on the probe shank? And this is the actual the future direction that we are currently working on this one, and that we can have multiple probe shank and multiple waveguide, and so we can switch the light from the back and completely packaged so that having a high density decoding site with the illumination of different colors switching from the same spot. As I mentioned, we wanted to have a capability having a precise control of each distal end of the illumination source by directly integrating light source onto the probe shank. And this is the micro LED, which is a neural dimension, 10 by 15 micron, and exactly positions surrounding the recording site. This one completely eliminating this packaging and assembly issues. But the issue here is the heating of this LED and it is lighting. And we make this probe by using a gallium nitride on silicon wafer. Thanks to this lighting business, there are a lot of blue LED research has been done. And typically on sapphire wafer, the recently technology has been developed in a way that they can resolve the lattice mismatch problem. They can integrate its quantum well on top of the silicon. So we acquire this quantum well grown structure, and then we can define a small geometry micro LEDs, and then we can etch the silicon by micromachining to release the probe structure. This is a fabricated structure, and we have a typical Bazaki configuration, 32 channel, 8 recording site for each probe, and then we put it 3 LEDs on each shank at the 60 micron separation. And this one is the grown-up view of a magnified view of the structure. This is the LED and meta, and the other one is the recording site. And this is the experimental data. We could successfully lighting these small micron LEDs and then release the structure very flat, and then we can manage the stress well. We can illuminate a single LED, and also we can illuminate a number of them. It has a pretty uniform illumination over the wafer and over the device. And this is a capability that I wanted to just show you and share with you. I'm not sure whether I can synchronize it with the music, which my students put it in this video clip. And let me try whether it works. I'm not sure whether you can hear the music, but I play with the music, but we can blink each LED by programming, and each LED can be illuminated at any site, any location by programming for the target region of the brain, and simultaneously you can record the signal. We put this the probe into the animal. I hope Yuri can talk about the recording site. Can we switch over to Yuri?
Sure. Okay, so this is
what Euisik mentioned is that optogenetics offered us something that we really needed. That is, we, meaning the circuit analysis people. Optogenetic has a very good or quite okay time resolution. However, it has a very poor spatial resolution. What I mean by that is, in a typical situation today, we put a fiber somewhere, we record somewhere else, and there is a distance of anywhere between 200 microns to a few millimeters between the light and the recording site. And in between these two sites, there are dozens, hundreds, sometimes thousands of neurons, and therefore we don't exactly know what we are doing because the circuit includes many, many different types of neurons. People who are working...
Can you make it in a slideshow mode so you can see the figure much larger? Slideshow, your screen?
It's on the...
PowerPoint slideshow?
It is.
Oh, really? Okay.
Can you see it? Do you hear me? Hello?
Yeah, we can hear you. Go ahead. I'm sorry. I interrupted you.
Can you see the screen?
Yes, we can.
Okay. So what I was saying is that we need a method where the light is delivered very close to the neurons that we are recording from at the same time. And this is what Euisik demonstrated, and I'd like to show you now how it works in action. This is a recording from a freely moving animal. This is the micro-LED probe placed in the hippocampal CA1 region. This is the site that is being illuminated, and these are the three recording sites shown here. You can see that out of these many possible neurons, a few of them only on the shank, which is being illuminated, is activated. And the reason for that is because the amount of light that we need now is in the nanowatt range. It's very small amount of light. So we can show with this nice sinusoid stimulation that neurons are entrained very close to here in the shank, and then we can switch to, let's say, to the shank 2, and then we can entrain neurons on shank 2, but not on shank 3, then vice versa, and this is the situation I showed you. In many situations, it is very important that we stimulate only neurons that we record from at the same time. So we will be perturbing neurons in the circuit only that we know exactly what's happening there. The goal would be, of course, given the ability to stimulate multiple sites, is to go down to stimulating only a single neuron. And this is something is illustrated here. These are four different experiments. This is the first experiment, where you can see that now this particular site is being illuminated, and there are only two neurons in the vicinity that are being significantly activated and is positively entrained. However, this is probably more than two neurons because we see a neuron in a neighboring shank that is inhibited. So there must be a circuit effect between here and here. Here's another situation that there are a couple of neurons weakly activated on this shank, and there is an inhibited one on the other one, and so on and so on. And we could do various combinations. Here's a illustration that indeed we can do better, and we can stimulate a single neuron on a particular shank. So now we are stimulating either the lowest or the top one, top LED, the micro LED. And what you can see here is when the bottom one is stimulated, there is a nice entrainment of this particular neuron, but not when the other LED is activated. Vice versa, when the top LED is activated, then the nearby neuron is being activated, but not the one which is about 200 microns away. So what we can summarize is that circuit analysis requires that we not only stimulate neurons that we don't know which types are, we don't know how many of them and what kind of circuit effects they will produce, but we have to stimulate only we should stimulate only those neurons that we can record from at the same time. And hopefully we can do it in a controlled manner. this can be done in a freely moving animal, and one of the main goals of this project that we are doing with Euisik is that every single thing that we do has to be performed in an animal that is moving around freely. The head is not fixed, so the real estate on the head will determine how many sites and how many silicone probes we can put on the animal. I'd like to emphasize also that this method can be combined with two-photon laser scanning microscopy, and it's sort of ready to be used. So what we heard from
You're looking at the display screen and you're showing us the screen used to form the figures. So right now we have a figure that's the size of a postage stamp. You need to flip the two. You need to flip the local and the display version of your PowerPoint.
What I do is I go to slideshow, and we checked it, and it was okay.
Wrong, but it's the other way. But you're looking at the slideshow, but you're showing us the underlying figures.
Okay.
Maybe what you need to do is lock the two screens together.
How do I do that?
You have a Mac or a PC?
Mac. Share. That's right. Share my screen.
Go to the display control, and then there'll be a window in the display control that says merge the screens.
Share my screen.
No, go to display in the control panel, and at some point they ask you whether you want to merge the screens, and you want to merge them.
So I need better instructions than that because.
Oh, sorry.
Maybe
what you could maybe just stop. Don't
okay,
do the
I don't want to waste time with this.
Because right now we're looking at very tiny figures.
Okay. I can show it from my side if that's not that's problematic. I can display from here while Yuri is presenting there. I can display from here.
Okay, that'd be great. Yeah, I mean we just
we just switch to me, and then so that Yuri can just talk and I'll display.
Okay.
But tell me, Yuri, when to flip the slide.
Okay.
All right. Sorry to interrupt.
Oh, thanks very much. I apologize.
Can you see it? Can you see it?
I can.
Yeah, you're on slide 1. Or 23, I guess.
Okay.
23 is okay?
Yes, perfect. Did you see this before, or should I start all over again?
No,
Matt?
we're fine. We got, okay. Yeah, just pick it up. Yeah.
Next slide. So this is the slide that is probably the most critical. There are two LEDs, micro LEDs of the same shank, and we can stimulate a single neuron either with one or the other LED. And this is where I started to conclude that this is a tool that allows us to stimulate single or very few neurons in a combination of with any time for a combination. The important thing is that every single neuron that we are activating with light will be recorded simultaneously. And you heard me before that the goal is to do this all in freely moving animals, and everything that we do in the laboratory is performed in a freely moving animal. So this is the summary. There are technical parts of it. Because we are delivering light locally, we need only tens of nanowatts of amounts of light as opposed to the milliwatts that people are using throughout the world. Obviously, this technology requires a little bit of tweaking, but it's ready to go. It's a fundamentally different approach than Changhuei is trying to do, of course, which is wonderful. This is invasive, but there's no light scattering issue. We go to the mountain, we go to the neurons very close, and this is how we try to tickle them.
All right.
Cool. Well, thanks both Yuri and Euisik. At this point, let's turn it over to Bernardo for any questions and discussion on these presentations.
Thank you. It's a very exciting technology, and I have two very practical questions. The first one is, as somebody who wants to use it, how close are you to mass production and getting it out there and commercialized? It looks very close.
That's a good question, and we are still working on it, and we have a second generation prototype, and we are currently working on third generation prototype, and that will be tested in your lab and also disseminated to the other laboratory. And after that, after we validate the probes in those sites, and then maybe that can be translated into other communities at the time.
And then, you already made the point that you're delivering the light locally, so you can use much less power. But on the other hand, now the light source is also very close to the electrode, to the contact site. And so how are the photoelectric effects, and what are the limitations imposed by photoelectric effects?
We're still analyzing that, whether photoelectric effect is really making a noise, or we also having a coupling noise. We are driving LED very close to the recording site, then also making a significant electrical coupling. So currently, our measurements show that there's no significant photo electric effect as compared to electrical coupling. So, which we are currently working on it to minimize it so that you can do in vivo recording without a signal processing significantly.
I see, very interesting. So even for high frequency light pulses and high frequency recording, no photoelectric effect.
Not significantly as far as we know, but a little more investigation has to be needed.
Great. Well, I look forward to getting my hands on one of these.
Euisik and Yuri, very nice talk. ANC has a safety limit on the irradiance or fluence rate, which is about 2 milliwatts per millimeter squared. Now, of course, the ANC safety limit is very, very conservative. I mean, you can go above it by about an order of magnitude without causing damage. But the numbers you quoted on some of your slides are actually pretty close to that number. I wonder if anyone has quantified heating in tissue.
We have done measurements about that in wild-type animals, and there is absolutely no indication, even when we are using long time, that there would be any heat-induced effect on neurons. This is a typical problem when you use a single fiber, which requires milliwatts of light. We are talking about micron and sub-microwatt light, and I don't think that will be an issue.
Right. So I think that the ANC actually regulates wattage per area. They don't regulate the total wattage because when you dump light, close, especially maybe near the fiber tip, your light beam is actually quite condensed. I wonder if there's any local heating that was monitored. When you're away from the fiber tip, I think light will be sufficiently scattered. So the fluence rate will be much, much lower when you're away. So someone probably has to measure the temperature right outside the fiber tip.
How about measuring the firing of the neurons? So that's much more sensitive than measuring anything there. And that's what we did with the wild-type animals, that no matter how you stimulate, you see absolutely no effect on firing activity of wild-type neurons, right, unless you have general rhodopsin inside.
And also we have some thermal modeling to estimate the temperature increase of the tissue, and we did some in-vivo characterization. But we don't have actually in-vivo measurement of actual temperature of the sensor at the illumination site when we have a light illuminated yet. But we think that it's pretty small and negligible because our model is pretty conservative, because we do not consider any blood circulation, which will also contribute to lower the temperature of the probe tip.
Right. What's the typical distance between the fiber tip and the channel rhodopsin?
Yuri may answer the question.
It can be anywhere between 5 micron or 50 micron, because this is the radius where our recording electrodes can monitor the spikes of the neurons. And we can tell how far the neurons are from the amplitude of the action potential. So if an action potential that we record from is about 200 microvolts, then we know that that neuron is within 5-6 micron from the recording tip. And from the recording tip, we know exactly how far we are from the LED, micro LED.
Right. Euisik, you also mentioned about you want to create some artificial memory. Would you elaborate on that?
What do you mean artificial memory?
Oh, I mean, so you use an array of excitation, right? Are you trying to form some pattern of memory? Did that make sense?
Absolutely.
That was in the slides that we got as a preview, but not in the slides that they actually presented.
We are testing currently, with the help of this technology, all the heavy and pairing paradigms that have been tested in vitro already, and see how it works in vivo. We can use any patterns. We can present noise patterns and see how the noise pattern can affect local circuit activity. We are inducing various patterns such as sharp wave ripples, data oscillations, and eventually, if you want, you can put in a pattern artificially that you can read out subsequently during sleep that you can call an artificial memory.
Right.
That's a secondary issue.
We are looking forward to that.
Very exciting. Thank you.
Cool.
Can I have one more question, or are we done?
Yes,
Sure, go ahead.
You mentioned that the GRIN lens gives you an advantage in the coupling, that you can be less precise.
Right.
I don't really quite understand that, because it appeared you were using the GRIN lens to do a one-to-one imaging relay. And so what's the advantage there as opposed to putting the waveguide immediately onto the micro LED?
Okay, we did it initially. The easy way is just a butt coupling. Put it together as close as possible. But we have a pretty good collimated light coming out from LED. But however, also we can put the index matching epoxy to guide the light into the waveguide. But our coupling, loss is significant, and also what we are talking about, we are assembling multi-SHANK, multi-LDs, and we have to maintain a pretty good yield of the assembly. And then also we do the simulation, which we submit a paper, the tolerance, alignment tolerance coming from GRIN lens versus butt coupling. And we have a pretty reasonable and good tolerance level coming out from GRIN, because the position of the LD at the end and misalignment of the GRIN doesn't really matter, because the GRIN allows us to put the light onto the center and they're coming out from the other end.
Okay.
Cool. Well, I'd like to go ahead and thank Euisik, Yuri, and Changhuei for presenting today, and Bernardo and Li Hong for serving as discussants, as well as everyone else for dialing in on the Friday afternoon here to listen to the presentations. As a heads up, we have decided to go ahead and cancel the one originally scheduled for July 1st, primarily due to scheduling conflicts and it being the holiday weekend, and look for the emails from Matt Raymond to go ahead and set up the next one. Once again, thank you all for attending, and have a good weekend.
Thank you very much.
Thank you.